Hello everyone, in this video we will discuss the Melaka paper 1 for the year 2021, and that's the objective paper, and this is part 2 where we will discuss questions 21 to 40 which are related to Form 5 topics. So in this video we're going to revise some Form 5 topics as we discuss the questions. Question 21. Diagram 12 shows the growth zones in a plant shoot. What is zone Y? So here we can see 3 different zones, which is zone 1, 2 and 3. Okay. Now zone 1 is this one. It has this portion here which at the very tip of the shoot, which we call the apical meristem. Okay, at the shoot tip. And so this zone is the zone of cell division. Whereas zone number 2 below the apical meristem is the zone of cell elongation, and zone number 3 is the zone of cell differentiation. Okay, so zone Y here, which includes the apical meristem at shoot tip, must be the zone of cell division. Okay, so it's C, zone of cell division. Usually we call it zone of cell division. Right, next question. Which of the following pairs of tissues and functions are correct? Okay, so let's have a look at this. Vascular tissue for transporting water and nutrients. That's correct. So the vascular tissues in the plant are the xylem tissues and the phloem tissues. Xylem tissues transport water and phloem transports nutrients. Okay, so number 1 is correct. Number 1 is correct. Meristem tissue controls the loss of minerals. So this phrase is not correct. The meristem tissue is to divide by mitosis, helps to produce new cells through mitosis. Okay, meristem tissue helps to produce new cells through cell division by mitosis. So number 2 is out. Number 3, mesophyll tissue provides support. Wrong. So the mesophyll tissue, like the Palisade mesophyll tissue and the spongy mesophyll tissue in the leaves of the plant, helps to carry out photosynthesis and produce the organic food substances for the plants. Alright, they carry out photosynthesis, so this is out. Epidermal tissue protects the tissues beneath it. This is correct. So the epidermal tissues are at the uppermost layer of the leaf, for example, right, and protects the tissues beneath it. So number 1 and 4, so the answer is T. Question 23. Diagram 13 shows a chloroplast in a plant cell. Which of the structures A, B, C or D is the site for light-dependent reactions for photosynthesis to take place? Right? So we know that for photosynthesis, in photosynthesis there are 2 stages. The first stage is called the light-dependent reaction, and the second stage is the light-independent reaction. Okay, so for the first stage, the light-dependent reaction, the site for this reaction is the thylakoid. So looking at the 4 labels here, A is the thylakoid. So this is the answer. So what is the thylakoid? Thylakoid is actually a disc-shaped structure which contains chlorophyll. So it's green in color. And this chlorophyll absorbs the light energy which is needed for the light-dependent reaction, where water is broken down into hydrogen ions and hydroxyl ions. Okay, so all that occurs in the thylakoid. Now, if there's a group of thylakoids here, okay, that are stacked up like this, we call that granum, a granum. So the light-dependent reaction occurs in the thylakoid or the granum, in the granum or grana, inside the chloroplast. Now, let's label the other parts. So B is the inner membrane. C is the stroma or the liquid part or fluid part in the chloroplast. So the stroma is the site for what reaction? Light-independent reaction, which is the second stage in photosynthesis. So stroma is the site for the light-independent reaction, whereas thylakoid is the site for the light-dependent reaction. Alright, now D is the outer membrane. Question 24. Diagram 14 shows a cross-section of a leaf. This plant is exposed to dust from a nearby housing construction site. Question. What is the effect on photosynthesis if structure X here is covered with dust from the construction site? So X is the layer above the epidermis. I think it's the cuticle, right? So waxy cuticle. So imagine that it's covered with dust from the construction site. Now the question is, what is the effect on photosynthesis, right? So we have to know the equation for photosynthesis, which is WAC-GO. Okay, this is our acronym, which is very useful for us to remember the process of photosynthesis, right? And for respiration is the opposite, GO-WAC. So now this one is photosynthesis, is WAC-GO. Now water, energy, and carbon dioxide, okay, they are used to produce glucose with the release of oxygen into the air. These are the products of photosynthesis, glucose and oxygen gas. So back to our question. Now we have to underline here what is the effect on photosynthesis. Photosynthesis, structure X covered with dust, like this. So if the waxy cuticle and also the lower layers are already covered with dust, it will prevent something from penetrating the leaf, and that is sunlight, correct? So sunlight cannot penetrate into the leaf, and thus these cells below, now what are these cells? Okay, now this layer is called the Palisade mesophyll layer. So each of these cells are the Palisade mesophyll cells. So these cells will not be able to receive light, right? Light energy for photosynthesis. Okay, so less light reaches Palisade mesophyll cells, and the rate of photosynthesis of in these cells will decrease, right? So rate of photosynthesis will decrease. Now let's look at the choices. The production of starch increases. So this is wrong, because if rate of photosynthesis decreases, less glucose is formed, and then less glucose will be converted to starch for storage. So it should be the production of starch will decrease, not increase. The production of glucose decreases. Is that correct? Yes, because glucose is a product of photosynthesis. Now the rate of photosynthesis has decreased, so production of glucose decreases. This is our answer. Okay, now let's look at C. The release of oxygen to the environment increases. No, as we can see from the equation here, the oxygen is a byproduct of photosynthesis. It should decrease, right? The amount of oxygen released should decrease because the rate of photosynthesis decreases. So it should be decreases, so it's wrong. D, the release of carbon dioxide to the environment decreases. Now carbon dioxide is not a byproduct of photosynthesis. It is a substrate, right? So there's no release of carbon dioxide. So this answer is wrong. So the answer is B. Question 25. Which of the following is the problem faced by a plant that is caused by lack of nitrogen? A, unhealthy root growth. Now unhealthy root growth is an effect of deficiency or lack of another macronutrient, not nitrogen, and that is phosphorus. Okay. B, the leaves or whole plant turns yellow. Again, this is caused by lack of another macronutrient, which is sulfur, right? The leaves have chlorosis and the lower leaves fall off. Chlorosis means yellowing of leaves. Yes, this is the symptom shown by lack of nitrogen. Okay, so answer is C. Now D, photosynthesis process cannot occur and there's stunted plant growth. Okay, this is caused by a lack of carbon, oxygen or hydrogen. Carbon, oxygen or hydrogen. All right, so the answer is C. You can check up the symptoms or the effects of deficiency in your textbook, okay, on this page. Question 26. Diagram 15 shows 3 types of plants, R, S, and T. What is the nutrient adaptation in plant R, S, and T? Or what are the nutritional adaptations, the different ways in which they obtain nutrients, right, for plant R, S, and T. Okay, so identify these 3 plants first of all. Now, plant R is growing on a tree trunk, okay, and it has roots, it has leaves, so it's probably an epiphyte, which grows on the trees to acquire a higher position so that they can get more sunlight for photosynthesis, right? But they are not parasitic. They don't absorb any nutrients from the plants, all right? They can carry out photosynthesis on their own. So they are epiphytes. Now, this plant, of course, we can recognize that it is Rafflesia. And Rafflesia is a parasitic plant, okay, that obtains nutrients from its host tree, okay? And then this plant is the Venus flytrap, which is a carnivore. It can synthesize its own food through photosynthesis because it has green parts, but it also traps the animals, okay, in these structures, and the trapped animals are digested to supply nitrogen to these plants for growth, because these plants usually grow in soil which lacks nitrogen, right? So they obtain the nitrogen from the animals that they digest for healthier growth, right? So let's look at the choices here. So the answer is B, because it fits what we have discussed just now. R is epiphyte, S is parasite, and T is a carnivore, meaning an organism. Carnivore is an organism that eats other animals, that eats animals, not other animals. An organism that eats animals, okay, such as insects, right, in this case, for the Venus flytrap. Question 27. The following information describes root pressure, which is involved in the passage of water and mineral salts from soil to leaves. Now, root pressure moves water from the soil into the xylem vessels of the root by osmosis. So root pressure is a force that will move water from the root hair cells into other root cells until it reaches the xylem vessels in the center of the root. And then the water will also diffuse from the root cells into the xylem vessels by osmosis. So this is how the force called root pressure is created, which helps to move water in the roots of the plant. Okay. Now, what factors affect root pressure? The water potential in the soil is lower compared to the root hair cells, or the water potential in the soil is higher compared to the root hair cells, or the water potential in the soil is the same as the root hair cells. So just imagine an epidermal cell of the root with the root hair to increase the surface area for absorption of water. Now, in the cell septum, all right, in the vacuole of this root hair cell or epidermal cell of the root, there are lots of dissolved substances like mineral salts, and thus the cell will have lower water potential, okay, or lower water concentration compared to the external environment, compared to the soil solution outside the root hair cells. So soil solution outside, we assume that, let's say there's rain, so it may dilute the soil solution, and it usually contains higher water concentration or higher water potential, all right, than inside the cell. So the soil solution has higher water potential than inside the cell, and that's how osmosis occurs, where water diffuses into the cell from the soil solution by osmosis. And then from this cell, this one cell, the water will diffuse to the next, to the adjacent root cell, and so forth until it reaches the xylem vessels in the center of the root. And that's how the root pressure force is produced. So the answer to this question, what factors affect root pressure or cause the root pressure to occur, it should be B. The water potential in the soil is higher compared to that in the root hair cells, and that's how water can diffuse into the root cells by osmosis to produce the root pressure force. Question 28. Diagram 16 shows water spinach used for phytoremediation, a method of treatment for the purpose of degradation, extraction, or elimination of pollutants from soil and water. Okay, so this is the water spinach, which river water spinach, which absorbs heavy metals like cadmium from water. So it is used in phytoremediation, a method of treatment to extract or eliminate the pollutants from water by using plants to absorb these substances, the pollutants like heavy metals, so that the level of pollution in the soil or river water or other bodies of water can be reduced, to reduce the level of pollution in soil and water. So what is another example of phytoremediation plants that is correctly matched to their function? This is a straightforward question. So we need to memorize all the examples of plants that are used in phytoremediation to reduce the level of pollution in soil and water, right? So let's read the answers. Hydrilla plants used to absorb the heavy metal cadmium. No, this is not one of the examples given in the textbook. Balsam plants used to treat wastewater in sewage plant. No, this is not true. Chrysanthemums used to absorb mercury from the soil. Now, chrysanthemums are yellow in color, and some people may mistake them for the sunflower. But chrysanthemums are not used to absorb mercury from the soil. It is the D. Sunflower plants are used to eliminate radioactive materials such as cesium. All right. So this is the correct answer. This example was mentioned in the textbook, page 90. Right. This is the sunflower plant, not chrysanthemum. So sunflower plants are used to eliminate radioactive materials such as cesium from the soil, right? Now these are two examples of phytoremediation that are studied from the textbook. And then there are two more. Water hyacinth or Econia species plant, which is a floating plant on the river, okay, top on the river surface. It helps to absorb and accumulate heavy metals such as copper and lead in water, so it will eliminate or extract these substances out from water. So it can reduce the level of pollution of river water, okay, and so forth. And then groundwater spinach helps to absorb mercury from the soil. Groundwater spinach grows on the ground, not in the river, and so it can help to absorb mercury from the soil. So there are 4 examples here that you need to know. Examples of phytoremediation. Question 29. Diagram 17 shows a coleoptile tip which is cut and placed on an agar block. Then it is placed inside a box with a hole and exposed to the light source. What is the observation of this experiment after a week? Okay, so here in this diagram you have to trace the movement of the auxin, which is the hormone that can stimulate elongation of cells, right? So the coleoptile tip is cut first of all. This part is cut and then placed on an agar block and then put back onto the coleoptile. So coleoptile is just a shoot tip, right? And at the very upper part here, this is the zone of cell division, all right, which produces the auxin, the hormone auxin. Okay, now can the auxin diffuse through an agar block? Yes, I can diffuse through the agar block, which is porous, and it can move down into the zone of cell elongation here, where the auxin will stimulate the elongation of cells, right? So the coleoptile tip functions to produce the auxin, the hormone auxin, and then hormone auxin has to diffuse downwards to stimulate elongation of cells in the zone of cell elongation here, right? And the other thing we need to know is that auxin moves away from light. Okay, so let's trace the movement of the auxin. Now, since the light is coming from the left side here into the box, so the auxin will move away from light this way to the right side, to the shaded side here. And after that, there's a higher concentration of auxin on the shaded side, and this auxin moves down to the zone of cell elongation, where it stimulates elongation of cells. So auxin moves away from light, and the auxin concentration is higher on the shaded side here, right? So in the zone of cell elongation, then these cells in the shaded side will elongate more than the cells in the bright side here on the left hand side. Thus the shoot will bend towards the light. It won't grow uniformly on all sides. So it will grow more on the shaded side and then on the bright side. Then this will cause the shoot to move towards the light. So the answer is C. Okay, the shoot will move towards the light as it grows. So here's a picture that shows the movement of the auxin more clearly. The light's coming from the left hand side, auxin moves to the right hand side, so all these red dots are the auxin, and then the auxin, there's a high concentration of auxin on the right hand side, which is the shaded side, and then the auxin diffuses down into the zone of cell elongation, so it will stimulate cells to elongate more on the shaded side than on the bright side. Thus, the shoot will grow towards light. Question 30. Diagram 18 shows an embryo sac of a flowering plant. What will happen to cell P here, right? So we have to recall back the formation of the embryo sac in the flower, in the ovule of the ovary of a flower. Okay, so actually the embryo sac is formed from one cell called the megaspore mother cell in the ovule, which is diploid. So this megaspore mother cell divides by meiosis once to produce 4 haploid megaspore cells as shown here, and these 3 megaspore cells below here degenerate, leaving only one cell, which is P, to develop. Okay, so P is a megaspore cell that develops. Then the nucleus in P divides mitotically 3 times. Alright, so the first time it divides, one nucleus will become 2 nuclei. Then the second time, the 2 nuclei divides to form 4 nuclei. And then for the third time, the 4 nuclei divides to form 8 nuclei. Right? So in the end, this produces a cell called the embryo sac, a big cell with 8 nuclei inside, as shown in the diagram here. Now this is a mature ovule in the ovary of a flower, and we can see there are 8 nuclei that have arranged themselves in this order. Some of the nuclei are enclosed by a membrane, so they form cells in the end, right? So 3 of the nuclei move to one end, they form the 3 antipodal cells, and 2 nuclei in the center here, they are called the 2 polar nuclei. Then below there are 3 nuclei that will group together here, and they are enclosed by membranes. So one will form the egg cell, and 2 more will form the synergid cells. And all these cells are enclosed inside the big cell called the embryo sac. Okay, there's no embryo here yet, but it's just called an embryo sac, right? Now, let's look at the answers here. What happened to cell P? Cell P will degenerate. No, it does not degenerate. It will develop. Then P cells will enlarge and grow to form 2 nuclei. No, it will form 8 nuclei in the end. The P cell will undergo 2 meiosis. Not true. It will undergo mitosis 3 times. Okay, so this is out. To produce a cell with 4 nuclei. Wrong. Now D, P cells will undergo mitosis 3 times. That's correct. Actually, it's the nuclei that undergo mitosis, not the cell. Okay, the nucleus in the P cell will undergo mitosis 3 times. It's the nucleus that undergoes mitosis 3 times to produce a cell called the embryo sac, which has 8 nuclei. So the best answer is D. All right, but take note, it's the nucleus that undergo mitosis, not the whole cell. Question 31. Which statements are true about formation of male gametes? Firstly, the generative nucleus will divide mitotically to form 2 haploid male gametes. Okay, so underline generative nucleus, mitotically 2 haploid male gametes. Okay, now this occurs when the pollen grain has landed on the stigma of a flower and it has grown a pollen tube downwards like this, in this picture. So inside at first there is the generative nucleus and the tube nucleus, right? The tube nucleus is near the tip of the pollen tube here, leading the way. Then what happens to the generative nucleus? Yes, it will divide by mitosis to form 2 haploid male gametes, okay, as in this picture. So number 1 is correct. The generative nucleus will divide mitotically to form 2 haploid male nuclei. Now, male gametes or male nuclei. Now, the second one, the tube nucleus will degenerate and both male gametes will enter the embryonic sac, right? So, when the pollen tube has reached the ovule, the tube nucleus will degenerate, correct? And both male gametes will enter the embryonic sac or the embryo sac. So that's correct. All right. Now, thirdly, the end of the pollen tube will secrete hormones to digest the tissues of the stud. So this word hormones is wrong. It should be secrete enzymes. Enzymes are not hormones. So it should be secrete enzymes, right? So this third statement is wrong because of the wrong term used here. Now, fourthly, pollen on the stigma will germinate and form a generative tube. So it's not a generative tube, it's called a pollen tube, as seen here, right? So again, wrong term used. So answer should be 1 and 2, so it is A. Okay. Question 32. The diagram, diagram 19, shows a type of plant. Which of the following explains the habitat of the plant? A. Swampy habitat which is rich in salt, such as in river mouth, the meeting point of fresh water and seawater. B. A habitat that's hot and dry with minimum presence of water, that is placed with high temperatures. C. A habitat that's not too dry or too wet with an adequate supply of water. And D. An aquatic habitat either on the water surface or submerged in water, right? So if you look at this plant, it's very easy to recognize that it is a rubber tree, right? And rubber tree is a mesophyll, right? Which lives in a habitat that is not too extreme. The conditions are not too extreme. Okay? So the answer should be C. A habitat that is not too dry, not too wet, and it has an adequate supply of water. Okay? Question 33. The following statements describe the main characteristics of a microorganism. No obvious nucleus because there is no nuclear membrane. So highlight this up. No nuclear membrane. Sizes range from 1 to 10 micrometers long, and it exists in single cells, as single cells, or pairs of filaments, chains, or groups. So this is another hint or a clue for us to know what type of microorganism it is. Okay, so what are these microorganisms? Are they protozoa, or bacteria, or fungi, or algae? What is your answer? Yes, the fact that it has no nuclear membrane tells us that it is bacteria. All right, because bacteria is classified as a prokaryote, which is a type of cell that lacks a membrane-bound nucleus. So the genetic material of the bacteria is not bound by a nuclear membrane. And this type of cell is called a prokaryote. So bacteria are prokaryotic cells. Okay, so answer is B. Whereas A, C, and D, they are all eukaryotes, eukaryotic cells with a nuclear membrane. Question 34. Diagram 20 shows the energy flow from producer to secondary consumer. What is the total energy passed down to the, now it shouldn't be secondary prima, it should be primary consumer, like in this Malay version, pengguna prima, to the primary consumer. Right. So here if you look at the diagram, you can see that the producer has 1,800 kilojoules of energy, and when the primary consumer eats the producer, then the energy is passed on to the primary consumer. Then for primary consumer, 60% of the energy is lost in excretion, 30% lost in respiration as heat. Okay, so the amount that's passed on to the secondary consumer will be only 100 minus 60 minus 30, right? So 100 minus 90%, which is only 10% to the secondary consumer. Likewise, from a producer to primary consumer, only 10% is passed on, right? From the producer to the primary consumer. Okay, let's read the comments here. 90% of the energy is dissipated into the environment through heat, living processes, and excretion for each trophic level. Okay, for each organism at each trophic level. So only 10% of the energy is transferred to the next trophic level, from producer to primary consumer and from primary consumer to secondary consumer. Okay, now what is the total energy passed down to the primary consumer? From the producer to the primary consumer. So we have to take this 1,800 kilojoules of energy, and you have to multiply it by 10%. Okay, only 10% is passed down to the primary consumer. The rest of it is dissipated, as we have mentioned here, into the environment. Okay, so our working will be 10%. So 10 over 100 times 1,800, and the answer is 180 kilojoules of energy. So the answer is D. Now, however, if they ask for total energy passed down to the secondary consumer, then you will have to multiply this value, 180 kilojoules, by another 10%. And that will be equivalent to A, 18 kilojoules. Okay, but since they are asking only for the total energy passed down to the primary consumer, which is this stage, so 10% of the energy from producer is passed on to the primary consumer. So 10% times 1,800 kilojoules of energy, and we get D as the answer, 180 kilojoules of energy. Now, question 35. The movement control order announced by the government to curb the spread of COVID-19 resulted in more time being spent at home and contributed to increased electricity consumption. Among the practices that can be practiced to reduce the use of electrical energy is to turn off the switches of electrical equipment when not in use. How can this practice contribute to environmental sustainability? So firstly, let's recall the meaning of this term, environmental sustainability. It means an environmental condition that remains the same without any reduction of natural resources, with assured quality of the surrounding environment for a long period of time. Meaning to say, it means maintaining the condition of the environment so that it is in a good state for a long period of time. Okay, so how does the practice of turning off the switches of electrical equipment, that means reducing the use of electrical energy, contribute to environmental sustainability, or contribute to maintaining the environment in a good condition? Now, A, reduce the cost of paying monthly bills. Now, this helps the household finances. It improves the household finances and reduces the expenses for electrical consumption, right? So, but it has nothing to do with environmental sustainability. So this is not the answer. B, reduce thermal pollution. So if we save electrical energy or reduce the use of electrical energy, does it reduce thermal pollution? Because reducing thermal pollution is one way to contribute to environmental sustainability. So does saving electricity contribute to environmental sustainability through the reduction of thermal pollution? I believe it does so, because water is used as a cooling agent to cool down the generators in the hydroelectric power generator plants, which are used to generate electricity. So if you use less electricity, then less water will be used as a cooling agent to cool down the generators. Okay, and less hot water that is produced will be discharged into the rivers or lakes. Less hot water will be discharged into the rivers or lakes after they have absorbed the heat from the generators. So the temperature of the river or the lakes will not increase so much, and this will reduce the death of aquatic life. Okay, so I believe B can be an answer, but let's look at C and D, where the answer is more obvious. All right, so C reduces the release of pollutants such as sulfur dioxide, and D reduces the release of pollutants such as carbon monoxide gas. Okay, let's see. Does reducing the use of electrical energy reduce the release of these pollutants? Yes, it does, because when you reduce electrical energy usage, so you reduce the burning of fossil fuels that is used to generate electricity. So fossil fuels like coal, they are burned to generate electricity, right? So if we reduce the use of electrical energy, less coal will be burnt to generate electricity, and this will result in less pollutants being released through burning of coal. So what are the pollutants released through the burning of fossil fuels or coal, such as coal? Pollutants are carbon monoxide, carbon dioxide, sulfur dioxide, which are released into the atmosphere and cause air pollution. So it reduces the release of pollutants such as sulfur dioxide. This is correct. And reduces the release of pollutants such as carbon monoxide gas. This is also correct. So there are a few answers for this question. The answer given is D, but C can also be an answer, and maybe B. Okay? Because other than carbon monoxide gas that is released, sulfur dioxide gas is also released from the burning of fossil fuels. Question 36. A student wants to estimate the population of snails in a garden. He catches 65 snails and marks them before releasing them. So all of them are marked. 65 snails are marked. After 3 days, he catches 40 snails and finds that 5 are marked. So what is the estimated population of snails in the garden, right? So now there's a formula for this, which is the estimated population of snails equals to number of snails or number of organisms captured in the first capture, okay, so that's 65 snails, multiplied by the number of snails captured in the second capture, which is 40 snails, and then divided by the number of snails which are marked, which is 5 snails. So if you do that, you will get the answer 520. That is, population size equals to 65, okay, the number of snails in the first capture, multiplied by 40, that's the number of snails in the second capture, and then the whole thing divided by 5. 5 are marked. That means the number of snails that are marked in the second capture is 5. So divided by 5. So you get 520, right? But if you forgot the formula for this capture, mark, release, recapture technique, which I call the CMRR technique, if you forget the formula, you can still get the answer by reasoning it out. And that is, out of the total population, which is P, 65 snails are marked, right? Okay, so 65 over the total population. Now this proportion equals to the sampling that you did after 3 days, where you captured 40 snails and 5 of them were marked. So in your sample, out of 40 snails, 5 were marked. So we assume that this proportion, 5 over 40, equals to 65 over P. And the second capture is a sampling that you are doing. So if you have this equation here, this line, so you bring the P over this side, right, in maths, and then you bring the 5 over 40 over this side, becomes 40 over 5. So 65 times 40 over 5 equals to 520, right? So you can either use the formula or you can reason it out by yourself if you forget the formula. Question 37. A married couple has different rhesus RH factor. The first child is an RH positive child. All subsequent pregnancies end with miscarriage. Determine the rhesus factors for the parents and the miscarriage fetus. So when we look at this condition, the clue here is that the first child is rhesus positive, and then all the subsequent pregnancies end with miscarriage. That means the second fetus onwards, probably the fetus dies and then it comes out from the uterus. So that's miscarriage. Keguguran anak, right? And also from here we find out that the married couple has different rhesus factor, meaning that one is probably rhesus positive, having the rhesus factor in the blood, and the other is rhesus negative, doesn't have the rhesus factor or antigen D in the red blood cells, on the red blood cells. Okay, so here when we look at the situation where there's miscarriage, it will lead to the conclusion that probably the fetus and the mother is incompatible in terms of the type of blood. That means The fetus is rhesus positive, but mother is rhesus negative. That's how the miscarriage can occur for the second and subsequent pregnancies. Okay, and then the father has a different blood type compared to the mother. So if the mother is rhesus negative, father is rhesus positive. So the answer is C. And we have discussed this situation, or we have learned about this incompatibility of the blood group from in chapter 10 of Form 4 syllabus. So here is the explanation, which I will just read through, and I will also discuss it in more detail in the next slide as revision. So mother has rhesus negative blood, and in the first pregnancy she has a rhesus positive fetus, right? So in the 9th month or during birth, fetal red blood cells containing antigen D or rhesus factor from the fetus enters the mother's blood. All right, then the mother's white blood cells will produce anti-D antibodies in response to the antigen D that has entered her body. All right, so she will produce antibodies because she's rhesus negative, right? So she'll produce antibodies to destroy the antigen D that has entered her body. But then these antibodies can move via the placenta back into fetal blood. And luckily for the first child, the concentration of antibodies is low, so it does not affect the first child. But in the second pregnancy, if the fetus is also rhesus positive, then again the mother is exposed to the antigen D or rhesus factor from the second fetus in about the 9th month. So she will again produce the anti-D antibodies in her blood. And this time, the concentration of antibodies is higher. So these antibodies move across the placenta and diffuse into the fetal blood. Then this time, because the concentration of bodies is higher, more red blood cells of the second fetus will be destroyed, right? Through agglutination and hemolysis. And this causes anemia in the fetus, and the fetus may die. The condition is known as erythroblastosis fetalis. Let's do a quick revision of this topic on rhesus factor from Form 4 Chapter 10. What is the problem in pregnancy that arises if the mother is rhesus negative but the fetus is rhesus positive? Okay, number 1, in around the 9th month, fragments of red blood cells from the fetus which contain the rhesus factor or antigen D may enter the mother's blood. Okay, so this rhesus factor is just a protein on some people's red blood cells. Okay, now when the mother who is rhesus negative receives the rhesus factor from the fetus, she will automatically produce the rhesus antibody as an immune response. Okay, because to her this rhesus factor from the fetus or this antigen D is a foreign substance, it's an antigen. Okay, so she produces the rhesus antibodies. Being rhesus negative, she produces the rhesus antibodies, and mom's antibodies will then enter the fetal blood through the placenta. Okay, because these antibodies dissolve in the blood of the mother and it can diffuse across the placenta. Now, for the first fetus, luckily, he or she will not be affected, all right, because the level of rhesus antibodies from the mother is low on first exposure to the rhesus factor, right? But what happens if the second fetus is also rhesus positive? Then this same process occurs where rhesus factor or antigen D in the second fetus red blood cells enters the mother's blood in the 9th month. Mother again responds by producing rhesus antibodies for the second time. Now this time, since it's the second exposure, the mother's antibodies will be produced at a higher concentration. All right, so then the level of rhesus antibody from mom or mother increases in second pregnancy, and then again it will enter the fetal blood. The antibodies will enter the fetal blood through the placenta. This time it reacts with the second fetus rhesus factor and destroys the rhesus factor. Okay, so the response will be now greater. There's agglutination or clumping together of the red blood cells of the fetus and destruction of the red blood cells of the fetus, and hemolysis, meaning destruction of the red blood cells of the second fetus red blood cells, all right, in the fetus. Thus, the fetus will suffer from anemia and it may die, right? So when it dies, then the mother may suffer a miscarriage where the fetus comes out from the uterus. That is called keguguran anak. This condition is known as erythroblastosis fetalis. Number 38, the diagram shows the variation of a trait in humans. So number of individuals, and then here is the trait, okay, against the trait, and there are two bars here. So which characteristic shows the variation? Presence of dimples. Is that possible? Now, first of all, you have to know that this type of bar chart can only be drawn for what type of variation? Is it discontinuous variation or continuous variation? Yes, it's for discontinuous variation. So bar chart where you have separate bars shows the discrete distribution, and it is only possible to plot it if the characteristic is discontinuous variation, shows discontinuous variation. Okay, so because for continuous variation, okay, where the differences are not distinct, then you don't get a bar, you don't draw a bar chart, you draw a histogram, like for number of individuals against the height of the person. But here it's not. Here is discontinuous variation because bar charts are drawn. Okay, so is it presence of dimples? Yes, it's possible because you have two bars. So the first bar may be for those people who have dimples, how many of them have dimples in the group, and the second one is how many don't have dimples, right? Now, how can it be blood group? No, because the classification of blood groups based on the ABO classification system, okay, means that there are 4 blood groups, right? So there should be 4 bars. Bar for blood group O, then A, then B, and then AB. Okay, but there are only 2 bars here, so it's not for blood group. This bar chart is not possible for blood group. And then C, skin color. Skin color shows continuous variation. So we cannot draw bars for skin color, for the different number of people with a different skin color. And also for weight, it's a type of continuous variation. So these 2 are out. So the answer is A. Number 39, how does Turner syndrome occur? A, non-disjunction of, there's an off here, non-disjunction of chromosomes occur in grandfather or grandmother. B, non-disjunction of X chromosome during metaphase 2. C, non-disjunction of X chromosome during meiosis 1. Or D, non-disjunction of Y chromosome during meiosis 1. Right? So first, we must know the meaning of the word non-disjunction. So non-disjunction means the failure of homologous chromosomes to separate during meiosis 1, or the failure of sister chromatids to separate during meiosis 2. Right? So if the homologous chromosomes fail to separate during meiosis 1, both chromosomes in a woman during the formation of the ovum, both the sex chromosomes XX in a woman will go to one daughter cell and none will go to the other cell, right? So this will produce an abnormal ovum with no sex chromosome because none go to the other cell. None of the sex chromosomes go to the other cell. So the ovum produced will have no sex chromosome, and the number of chromosomes is 22 plus 0. That means no sex chromosome X, right? Because a normal ovum should have 1 sex chromosome X. Okay. Now, if this abnormal ovum, 22 plus 0, with the total chromosomes 22 plus 0, is fertilized by a normal sperm. So normal sperm can have 1 X chromosome or 1 Y chromosome. But in this case, we take the normal sperm with 1 X chromosome, 22 plus X. So when these 2 gametes fuse together, the abnormal ovum and the normal sperm, we will get an individual with 22 plus 22, so 44 autosomes, and then 1 sex chromosome, so XO. And this produces the child with Turner syndrome. Okay, as seen in this picture here, abnormal ovum with no sex chromosome, 22 plus 0, fuses with a normal sperm with 1 sex chromosome X. Then Turner syndrome child is born. Okay, so among the answers here, the best answer is C, non-disjunction of the X chromosomes during meiosis 1 in a woman, in the formation of the ovum. Now, why is it not D, non-disjunction of Y chromosome during meiosis 1? we don't have two Y chromosomes where both go to one pole, see? So we cannot say non-disjunction of Y chromosomes. So we take the case of non-disjunction of the two sex chromosomes X in the woman, and then you can get the answer C. Right, the last question, number 40. The following statement describes genetic engineering. Recombinant DNA technology has successfully produced many beneficial varieties of crops such as rice, corn, and soybeans, as well as livestock such as cattle and goats. Genetically modified food GMF possesses DNA from other species of plants or animals. So which of the following is the possible disadvantage of genetic engineering, right? It endangers the natural species. Yes, this is possible, right? Because if farmers choose to plant the genetically modified crops like genetically modified rice in the future, then the natural species of rice will not be planted anymore, and then they will slowly become extinct, endangered and then extinct, because nobody is going to plant the natural species of rice anymore if farmers choose to plant the genetically modified rice in the future, which may have more beneficial qualities than the natural species. Okay, so this is possible. A is the answer. Now the rest are not disadvantages. Some of them are not disadvantages. For example, overcome worldwide food shortage. This is maybe an advantage, okay, of genetic engineering. Reduces price of food, thus increase food availability, and reduce problems of crops related to pests. All these could be related to advantages of genetic engineering. So here we want the disadvantage. Right, so A is the answer. That's all for this video. Thanks for watching, and do share, like, and subscribe. Goodbye for now, and all the best for your coming SPM examination.